my dearest little champions out there. Let's see if it is live or not. Just give me a quick second. Is it live? Is it not? Those of you watching, am I audible? Yes. It's finally live. And a very good afternoon to all my dear children. So those of you who had their exams today, how was it? Was it good? Was it bad? Was it okay? What's the scenario? We'll just wait for some more time so that everyone else can also join. So my dear children, when do you have your biology examination? Is it happening tomorrow or day after tomorrow? So keep on telling me that until we officially start this live really soon. Okay. So, those of you who are not able to reply or text over here, basically make sure you're subscribing to the channel because it's on subscribers only mode. So, Santosh, tomorrow exam biology, oh my god, okay, then we have to prepare nicely, right? Wishing you all the best. And Anand says it's on 15th of October, coming really soon, right? Okay, 2 more days. So, children, we will wait for some more time. And please share this link with all the other fellow biology students out there if at all you have any friends. Mohan says it's tomorrow. Sru says tomorrow is English. Okay, but I'm glad you're here. Okay. So my dear children, so today's challenge is again to solve 40 most expected questions with me. So those of you who have been with me in the part 1 video which we took on Saturday, this is going to be a continuity of that. But over here we are going to focus on the most high weightage chapters such as molecular base of inheritance, principles of inheritance and variation, human health and diseases, and also evolution. So there are 40 questions. And these questions are something that I have picked up from the question bank. Some of the questions are from the model question paper of the previous year. So make sure you are lively over here. And I just have one request to you. That is, please give the answers for the questions that I'm going to show you over here before even I do. So Gagana says, ma'am, midterm exam marks is 64. Thank you so much. I'm super proud of you, Gagana. God bless you. And you have done so great, which is why you have got that marks. Kudos to you. Chalo, let's start then. All of you who are accepting the challenge, click on the like button right now and tell me I am ready. Then I'll start off this session. Say I am ready, and then we will begin off the live session right over here. As I said, we want 100% participation, okay? So no one is saying that I am ready, which means that no one is ready. Ma'am, important diagram, Santosh, I would be giving a video today. It will be, I have already recorded, it will be given to you. Sru says I am ready. Okay, one person says I am ready, and I am just relying on that one person and going ahead because I know all my dear other children are gonna come along the flow. So the first question is here: the number of nucleotide pairs present in the DNA of the primary oocyte of a newborn. Remember, it's a primary oocyte and it's of a newborn. Anand is also ready. Mohan Acharya is also ready. Come on, tell me the answer for this question. Number of nucleotide pairs present in the DNA of primary oocyte of a newborn in human being. A, B, C, D. So how do you tell me the answer? Ma'am, 1A, 1B, 1C, whatever is there. Okay? So you have to tell me like that. So those of you who said we are ready have to give the answers as well. And my dear children who have not subscribed to the channel yet, subscribe, then only you will be able to write down the answers. Ananda says it's 1A. Okay? Anand says it's 1A. Is anyone else there to challenge Anand and say no, it's not 1A? As I said, we will take this live full swing only when you guys participate. Otherwise, it's going to be so boring for me and for you. Okay. So, answer is not A. Why it is not A, my dear children? You are asked about primary oocyte. So if at all we have studied what you call human reproduction chapter, no? In that we know that first we have the oogonia out there. Oogonia is having 2N, that is diploid number of cells. Now some of this oogonia, they undergo differentiation or maturation. They are not undergoing any division. They are undergoing maturation, and now they are transforming into primary oocytes, my dear children. So primary oocyte is also diploid in nature. 2N number of chromosomes are there, right? So generally in diploid we have 6.6 into 10 to the power 9 base pairs. If at all it was haploid, it would have been 3.3. So the second thing, secondary oocyte, if at all your question is asked like that, then it would have been 3.3. Hence your answer is B. Okay, it is B. I am getting other options, D. Okay, it's okay, my dear children. For the first question, I forgive you guys. Just a second. Even I have marked it wrong, but please don't mind. It's B. It's B, not A, okay? It's B, not A. IAS Dreamer Abhi says tomorrow is biology examination. You are going to rock your biology examination. And Dhanush gave me the correct answer. The answer is B. And over here is the picture, my dear children. 2N is where your primary oocyte lies. Now we are going ahead with the next question. Question number 2. And try to solve it properly. Take your time, solve it. Okay? A DNA segment has a total of 1000 nucleotides, out of which 240 of them are adenine containing nucleotides. So 1000 nucleotides are there, and out of that I have already told adenine is 240. Ma'am, what do I have to find out? I have to find the number of pyrimidine bases. What are pyrimidines? Pyrimidines are of two types. One is T, that is thymine, and the other one is C, that is cytosine. How will you find that out? Prakriti has given me the answer. It's 2A. Is it 2A? What about others? Do we agree with Prakriti's answer? So those of you who are getting wrong answer, let me tell you, this is a live session. You give wrong answers. When you give wrong answers, you will get to know what are the correct answers as well. You learn from your mistakes. That's not a problem. I am not here to judge you. We are friends. Okay? So keep on guessing, but do the question and then only guess. So 2C is your answer for this particular question. Someone says it's 2C. Fine. Please upload 5 marker, 3 marker important questions. IAS Dreamer Abhi, dear, I have already uploaded for each and every chapter. Can you please watch it? 1C. Okay. Chalo, over here, 2C. So everyone is saying C, which is why I have to get into this particular question and solve it for you guys. Listen, 1000 of them are there, nucleotides are there. Adenine is how much? Adenine is 240. Adenine is best friends with what? Thymine. Adenine and thymine are the ones that makes base pair. Guanine and cytosine are the one that makes base pair. So if 240 adenine is there, that means 240 thymine is there. Agreed? Now 240 plus 240 is equals to what? It gives me around 580. Sorry, 480. Not 580. Just a second, okay? It would be 480. Now, out of 1000, 480 of them are A and T. So 1000 minus 480 is equals to what, my dear children? Is equals to 520. Agreed? Now this 520 I have to divide between what? G and C. So 520 which is there divided by 2 is equals to what? 260. So 260 here, if guanine is 260, cytosine is also 260. Now if you are asked about the number of what do you call pyrimidines, how do you calculate? 240 plus 260, your answer would be 500. So your answer is B. Not C, not D, not A, it's B. Did you get it? How the answer is B? Oh, it's D. My dear children, are you getting it? How it's D? So initially it was 1000 nucleotides, right? Out of which it is clearly mentioned adenine is what? 240 of them. Now if adenine is 240, adenine's best friend, that is T, thymine would also be 240. Now 240 of them are being covered. Out of 1000 we know almost 480 of them are these guys. Now what are we left with? How do you calculate? 1000, it's not 100. 1000 minus 480 is equals to what? 520. So 520 of them are there. Now 520 of them which are there, I have to divide into 2. Guanine will be 260, and if guanine is 260, cytosine is also 260. And you are asked what is the number of pyrimidine bases in this segment. So pyrimidines are what? Thymine as well as cytosine. So 240 plus 260 is equals to 500. Okay? Now did you get it? How B? Prakriti, I hope it's clear now. Hajira is there. Hi dear. Ashraf is asking, ma'am, this question will be sure question. Listen, these questions are the most probable questions. These have been taken from the question bank. At least you will understand the concept. There are chances it might pop up exactly like that. There are chances you will get similar concepts as well. Okay? Aruna, is it clear, dear? Can I move ahead with the next question? Children, if I can move ahead with the next question, give me a thumbs up. If you want me to explain, ask me that, ma'am, can you re-explain it? I'll definitely do that. So those of you are saying it's done. Prakriti says yes. Aruna, do you need an explanation? I can do that if at all you want. Anand also says it's done. Okay. In that case, I think we will move on. Fine. Done. Okay. So answer is 500. I hope. Yeah, Aruna is also saying yes. Cool. Now, the next question. The two nucleotides of DNA are linked through 3 and 5 carbon of pentose sugar. By what bond? By what bond? The two nucleotides of DNA are linked through 3 and 5 carbon of pentose sugar. So, pentose sugar is this. Okay. 1, 2, 3, 4, 5th carbon is right over here, and then again you have a pentose sugar over here. 1, 2, 3, 4, 5th carbon is over here. So 3rd carbon over here and 5th carbon over here is bonded through what is being asked over in this particular question. Sudeep, I have already done chapter wise 2, 3, 5 marker. Can you please watch it? Okay. And those of you giving me the correct answer, kudos to you. Okay, Prakriti says it's 3B, kudos. Okay, Anand also says it's B. Monisha says it's B. Prakriti says phosphodiester, awesome, awesome. And I am Sru also says it's B. Santosh also says it's B. Yes, you guys are correct. The answer is phosphodiester bond. So over here, my dear children, the 5th carbon is attached to phosphate group. Okay, similarly, the 3rd carbon is also attached to phosphate group. There is an oxygen in between, which we do not mind usually. So anyways, this bond is known as phosphoester bond, and since it is happening twice, you call it phosphodiester. Di stands for 2. Phosphodiester bond. So this is your answer. Okay, so for those of you who gave B as the correct answer, kudos to you, and this is how it looks. These two bonds together, you call it phosphodiester. If at all you are asked, what is that bond between the 5th carbon and the phosphate group alone? Then it is phosphoester bond. Since it is happening twice, it is diester. Okay, next question. Try this out. GGU, GGC, GGA, GGG all code for glycine. Which salient feature of the genetic code does it prove? Try it out. Okay, try this question out. Aruna, you want me to re-explain? I'll come back to the question, okay? Just a second. For everyone else who is trying this, can you guys do one thing? Whenever you solve a question, can you say 4, A, B, C, D? In that case, I would be able to understand is which question you are wanting me to solve. 4. Okay, 4. Prakriti says the answer is B. This feature is known as degenerate codons. So I have Prakriti says B, Abhilash says it's D. Okay, I have mixed answers coming in. You keep on answering, I'll explain it to you. So over here, it says that different codons are there, but all of them are coding for glycine. All of them are coding for single amino acid. This is known as degeneracy of codons, or this feature is known as degenerate codons, my dear children. When more than one codons code for an amino acid, you call this degenerate codons. Okay, when more than one amino acid, when more than one codon codes for an amino acid, you call this degenerate codons. On the other hand, if at all you said that GGU is coding for glycine, what feature does it show? Nothing else is given. This is the only thing given. GGU codes for glycine. What feature does it show? It shows unambiguous nature of what you call codons, because codons are specific for whatever amino acid they code. But over here, multiple codons are given and one amino acid given that show degenerates feature. So this is what? B. Listen, a question can be coming up from this. That is the features of codons. So make sure you're going through the entire features really well. Okay? Ma'am, can you explain slowly? Yes, definitely I can. Okay, so your answer for this question would be B. Now, I would just go back to the previous question since you were asking. Aruna, listen to me carefully. Here it is asked 2 nucleotides of DNA. This is one nucleotide, this is another nucleotide. Okay, it is linked to 3rd carbon and the 5th carbon. What bond comes in between? So over here, this bond where the carbon is bonding with the phosphate and another carbon is also bonding with phosphate. Okay, this bond which is there, you call this as phosphodiester bond. If you're taking only this part, the carbon and the phosphate, it is known as phosphoester bond. But since it is happening twice, you call this phosphodiester bond. I hope it's clear, Aruna. Okay? Now going ahead with the next question, please make sure if at all you want, you can take a screenshot of this. Otherwise, you'll also get the PDF. Next question, aminoacylation of tRNA occurs during what? Aminoacylation of tRNA occurs during what? Transcription, translation, regulation of operon or DNA fingerprinting. Come on, my dear children. So those of you who got the correct answer, let me read out their name. Mohan Acharya and Lakshmi, Monisha, all of you got the correct answer. Even Prakriti got the correct answer. Those of you got the wrong answer, it's totally fine. Keep on going, keep on grinding. You will be able to do that. You guys know you're awesome. If at all you do not know, remember that. Okay? Yes. Aminoacylation of tRNA occurs during? Come on, come on. 5th question answer, 5 A, B, C, D. Aruna says it's B. Okay. Prakriti says it's B. Fine. We have 2 people saying it's B. Whether it's correct or not, let's check it out. So over here, aminoacylation of tRNA is also known as charging of tRNA. But you know what happens? The tRNA looks like this. There is a 5' end and there is a 3' end. Oh, just a second. Okay. There is a 3' end. On this 3' end, an amino acid comes and binds. This is known as aminoacylation. And this loop is known as anti-codon loop. It will go and bind on the codon of an mRNA. So whatever codon is over here, depending on that, there will be another anticodon over here. And depending on that, they would be having one amino acid over here as well. Okay? So, which is why your answer for this question would be translation. In translation, you need tRNA for translating all the information which is there in the mRNA. Fine? So, your answer for this question is B. Children, all of you think it's going on really fast? If at all you guys think I'm going really fast, please let me know. Yes, I can definitely repeat. Okay, listen to me carefully. Over here, tRNA is there. The tRNA's job is to what? Translate the information which is there on the mRNA. So mRNA will be having codons, for example, A, U, G and so like that. And here comes a tRNA with an anticodon loop over here. If A is over here in tRNA, it will be U. If U is over here, it will be A. If G is over here, it will be C. But when it comes with an attached amino acid depending on the codon. So over here at the 3' end, an amino acid will be joining. So over here, since AUG is there as a codon, the first codon, it will be having methionine as an amino acid. So this process of attachment of amino acid to the tRNA is known as aminoacylation. You also call this as charging of tRNA, and it is an energy requiring process. That means you need ATP for that. I hope it's clear now. Okay, so your answer is translation. Can I move ahead for the next question? Children, give me a thumbs up and I'll move ahead with the next question since there is a clash between. Okay, Aruna, bye-bye. Fine. Let's go ahead then. If a processed mRNA of a particular cistron contains 636 base pair that codes for a polypeptide chain, what are the number of amino acids that it codes for? Listen, this question is tricky. You have to be careful while you solve this question. Okay, so check. If a processed mRNA of a particular cistron, that means of a particular unit, contains 636 base pairs, 636 base pairs are there. That codes for a polypeptide chain. What are the number of amino acid is being asked? So if these many nucleotides are there, and if you already know that nucleotides basically they come in a group of 3. They come in a group of 3 to form codons. So it would be 2, 1, 2 codons. And if we know the number of codons, we also know the number of amino acid. So you might be thinking 2, 1, 2 amino acids will be there because each codon will be coding for an amino acid. But you know what? It's a cistron. It's a unit. And in the cistron, the last codon which is there is a stop codon which does not code for any amino acid. So 212 codons, if at all is there, you have to assume the last codon is not going to code for anything, which is why the number of amino acid is going to be B. Okay, it is going to be B. Fine. Yes, Aruna, it's totally fine if it's wrong. You get to learn something over here, so don't be disheartened, okay? And kudos, Prakriti going on really strong. Where are the other children? Everyone who is watching this video, please understand, this video is on subscribers only mode. So if at all you are not subscribed to the channel, you won't be able to answer it. Yes, Lakshmi, it is 5B. Kudos. Now let's boost up the energy, okay? So this is 6th question, and the answer for the 6th question is B, 211. Where is the last codon attaching then? So last codon is there. It is not coding for any amino acid. That's the only thing. That means there is no other tRNA that will come and join on the mRNA for translating the last codon. The last codon does not code for anything. It is known as stop codon. UAA, UA, UGA, and UAG. These are the 3 stop codons. If at all you see these codons, that means they're not going to code for anything. Cool? Okay? Fine, let's go ahead with the next question then. So I have written down the answer over here. Is it freely suspended then? Yes, it is. So there is an mRNA. The tRNA reads until this point. UAA is not read. Whatever amino acids have joined, that's all. And then later this moves out, that's all. Okay? Hello Sagar, long time. Really good to see you over here. Question number 7. In pea plants, each character is governed by a gene which is. Children, in pea plant, each character which is there, it is governed by a gene which is monocistron, polycistron, operon or intron. What is your answer for this question? Come on, let's see all of you answering. Aruna, kudos. Okay, Prakriti says it's 7A, monocistron. Okay, is there anyone to challenge Prakriti and say it's no, this is not the answer? Sagar, there will be free videos for KCET, do not worry about it, okay? We will definitely restart it. Okay, my dear children, C is the answer for this question, is it? So over here, it is mentioned pea plant. Listen, pea plant is what? A eukaryote. Eukaryote means the ones that has nucleus and also the nuclear membrane around it. Right? And wherever eukaryotes are there, they have monocistrons. Monocistrons means there will be one unit coding for one particular gene. Rather than in bacteria, which are prokaryotes, my dear children, they are polycistronic in nature. That means, suppose there is a one unit, over here there will be a gene, second gene, third gene in the same unit. That is known as polycistronic. So this is monocistron, which you find in eukaryotes, and this is polycistron, where you find multiple proteins being coded within the single structural unit. So which is why your answer for this question would be A. Okay, this is A. In pea plants, this is a eukaryote, and in eukaryotes, as I said, it will be always monocistron, not polycistronic. So those of you are saying C, Baba, it's not C, it's A. It's A. Okay, I hope you got it. Can we go ahead then? Fine. Chalo. Match the following. Next question out there which you can try to solve is this. Alec Jeffrey, F. Sanger, Jacob and Monard and Griffith is there, and these are the options that you have. What did they find out? You have to tell me that. So before I do, you have to let me know. So make it fast. Okay, now from this point onwards, children, I need everyone to participate really well. Listen, this is your examination that is going to happen tomorrow. So whenever you give the answers, you get it wrong, it becomes all the more in your memory that, okay, I got it wrong, I have to make things right. Okay, now I see the vigor out there. Prakriti says it's C, and then I have Mohan who says it's B, B, C, B. Okay, a lot of children have different answer for this, but C and B is where the competition lies. So I'll tell you easy ways to remember this. Alec Jeffrey, he has 2F. Please remember he is responsible for DNA fingerprinting. Okay, Alec Jeffrey for DNA fingerprinting. Can I go with that? Okay, A with 4. Now next, F. Sanger. S is for DNA sequencing. So annotated DNA sequencing was done by F. Sanger. Jacob and Monad, both of them basically has O in them, and it is they were responsible for finding lac operon. Griffith, as we all know, he is there for transforming principal experiment with the help of Streptococcus pneumoniae. So you can remember like that. Okay, but there is double F over here, do not be confused. Well, I think for Griffith, everyone can remember 2. Rest everything else is kind of difficult for remembering. So, yes. So, in that case, D goes with 2. Actually, there is only one option like that, which is why your B answer is the correct answer. Okay. So, those of you who got B as the correct answer, kudos to you. Those of you didn't, it's totally fine. We are on the verge. So, Lakshmi, Mohan, Dhanush, Sagar, Prakriti, all of you, kudos. Okay. Now going ahead with the next question that is there. Human insulin is produced in E. coli cells by rDNA technology. This proves that genetic code is what? So listen, as of now, this is not subscribers only mode. Those of you who want to participate can participate. Okay. Please make sure you are putting down the correct answers out over here. In human insulin, which is actually ours, okay, it is produced in E. coli by a technology that is rDNA, recombinant DNA technology. This proves that genetic code is what? Okay, Prakriti says it's A, Sagar says it's A, Dhanush says it's A. Okay, 3 people saying it's A. Akash, hello. Good evening. What do you think is the answer? You are saying it's A, which is why I'm locking A, and I'm really proud of you guys because the answer is correct. So, an insulin, which is human insulin, if at all it is produced in E. coli, that means genetic code which is there, it has to be the same, which is why the same thing is being produced. Okay, so this shows the universal nature of the genetic code. Okay, now going ahead with the next question out there. 10th, try to solve it and I'll agree that you guys are champions. Anyway, you guys are champions, but anyway, let's see. Who all knows this question? We have solved it so many times. Akash, once he got in, he gave the correct answer as well. Kudos to you, my dear child. And Sagar says, ma'am, there is English tomorrow. Okay, then I'll recommend you to go study English first if at all you think that you have not covered English. Okay, if you think it's covered, you can be here. Rishi also says it's C. Okay. Prakriti says 10th answer is C. Akash says it's C. Rishi says it's C. Let me see if at all you are correct or not. Well, since you all say it's C, I will also go with C and I'll give you an explanation for that. Listen, over here, the question is very simple. 37.2% recombinant Drosophila progeny, which was there, obtained in the experiment, my dear children. He said that there were phenotypes with red eye color, normal body, and white eye color, miniature body. This is due to, you don't have to focus on anything else, just on this number, 37.2. That's a huge amount of recombination. You know, 50% is the highest amount of recombination. So over here, suppose this is a trick that you can remember and I'll tell you it is going to be helpful for you. Here it is small a, small b. Here it is capital A, capital B. The genes which are present over here, what do you think? They are very close to each other. They are very close to each other, which is why I'll call them tightly linked genes, since they are very close to each other. And the distance, as you can see, is very, very less, right? Now, suppose the chromosomes, they get interchanged. The part of the chromosomes, they get interchanged. This will go there and this will go over here. But even if that happens, my dear children, capital A, capital B will still be together. Small a, small b will still be together. Why? Because they are tightly linked. That means the chances of formation of new combination is very less. So the recombination is not going to be this much percentage. It can be 1.3, 1.7%, things like that. On the other hand, think of another scenario where what is happening? Over here you have small a, over here you have capital A, over here you have small b, and over here you have capital B. These genes which are very far from each other, they are known as loosely linked genes. They have very much space between them. They are loosely linked genes. And now, if at all this part of the chromosomes get interchanged, my dear children, the top part, what will happen? Small a which was with small b will be now with capital B. Similarly, capital A which was with capital B will now be with small b. So there are chances for newer combinations to form. So here 37.2% recombination means what? They has to be loosely linked and there should be longer distance between the genes, which is why your answer would be what? Your answer would be C. I hope you got it, and there's a trick that you can remember. So if at all you and your best friend are really close to each other, there is no chance someone else can be your best friend. Okay, there is no chance new friendship can occur. So no new recombinations out there. But you and your friend, if at all you have so much space between you guys, then obviously there is a chance a new combination can happen. In that way you can remember. I hope it's clear. Okay. Now going ahead with the next question, and you want to read the question. By that time, I will read out the names of the children who attempted this. Okay, so I see Rishi, Akash, Prakriti, Sagar, Lakshmi, and Dhanush giving me the correct answer for this question. And yes, Sagar, what is the duration of this video? I do not know. We have to complete 40 questions. So that is the challenge that we have, and I hope you all have taken up the challenge. Now solve this question, my dear children. In a dihybrid cross in pea plants, Mendel got 9:3:3:1 phenotypic ratio. It denotes that. It denotes what? There is 9:3:3:1 ratio. You know what this means? He started off with crossing two type of plants, round and yellow and wrinkled and green. But at the end, you know what he got? He got round and yellow. 9 of them were round and yellow. 1 of them was wrinkled and green, and 3 were new combination. Here round was with green instead of being with yellow, and here also wrinkled, which was with green, was now with yellow. You found new combinations. Why? Because the alleles which were there for both of these genes, my dear children, were assorting independently. I see new people came in. Okay, so you all are saying that the answer is D, which is the alleles of two genes are segregating independently, and you guys are correct as well. Okay, do you have a doubt in this question? If you want me to explain, I can definitely do that. Or if at all it's okay, no explanation needed, I will move on to the next question. Just give me a thumbs up. If you need an explanation, I can give you. If you want me to go ahead, give me a thumbs up. Lakshmi also says it's D. Okay. Prakriti says, okay, gave me thumbs up, so I'll move ahead. My dear children, you can stop me at any point, write down the question number if at all you have any doubt, okay? Okay, fine. So before I go ahead, very important thing, that is about KC90. Do you know what is KC90? It's a crash course that we are launching. We cannot actually say it as a crash course. It's a course that is going to help you really, really well for your KCET as well as COMEDK preparation. It's a 90-day course, and we initially thought that we would take it offline, but as of now, seeing your response, we thought we should be making it online and making it easily available for all those children who won't be able to come to Deeksha campuses. But there are options. You can go for offline option at Deeksha campuses, or probably you can go for the online option. If at all you want any more details, you can call to this number or look out for the link given in the description box as well. Okay, fine. Now going ahead with the next question, question number 12. A pedigree chart is given below. Identify the trait responsible for this inheritance pattern. What do you think is the answer? Now while you solve this question, please make sure you're going through each and every step properly. I'll give you a clue. If at all it's a dominant disorder, you will find this in every generation. If at all it's a recessive disorder, you will find that it is skipping certain generation. That means parent won't have, but the child have, like that. So what would be your answer for this particular question? Okay, I see Prakriti, Dhanush saying it's 12B, 12B. What about others? So 22 people are watching this video and now they are not attending the answers. Oh my God. Is our confidence level that low? We should not be there. We should not be underconfident, my dear children. You should be super confident. Keep on answering whether it's wrong or right. 12B is what 2 people said. Apart from that, Yashwant also says it's B. Okay. So autosomal recessive. Why do you think it's autosomal recessive, my dear children? Because over here, first of all, why recessive? Because here, as you can see, both the parents are not affected. That means it is skipping generation. But the children are. Again, here the parents are not affected, but the children are. So this is a recessive trait. Now, X-linked. Why it is not X-linked and why it is autosomal? That is again a question. If at all, my dear children, it would have been an X-linked disorder, you would have seen that from the father, it would have passed to the daughter and then to the son. You do not see that pattern over here. And mostly, here the male is affected, here the female is affected, here the male is affected. It's not like the males are the ones which are mostly affected. I do not see this male, this male, this male, everyone to be affected, right? If at all, the males are affected more in number, then we would have thought of what? X-linked disorder. And since it is not, we can go for this particular thing, that is autosomal recessive trait. Okay? So your answer for this question would be B. Yes, Prakriti, because both female and male are almost equally affected. Yes. Now let's go ahead, and before I read out the names, you can solve this question. So I have Prakriti, Dhanush, Yashwant, Bhavya, Shri, hi Bhavya, and Lakshmi who gave me the correct answer, and Alan is back over here who has given the correct answer. Kudos to you. Now going ahead with the next question, which of the following is not a recessive gene-linked disorder? Recessive gene-linked disorder. 13A is what? Okay, 13A, Yashwant is also saying A, Alan is also saying A. Anyone else to say that they are wrong? Anand says it's A, okay. I see the number of people increasing. Kudos to all of you. I am super proud of you guys. I am super proud of your participation. So over here, the answer is actually A. Why? In your NCERT, I'll tell you, all the diseases which are given are recessive disorders. It can be sex-linked or it can be autosomal, but it is mostly what? It is recessive disorders. Only one disorder is given, that is myotonic dystrophy, and that too in a pictorial format. So if at all you have seen this picture, my dear children, in your NCERT, this pedigree chart which is given is of myotonic dystrophy, and at the top it is written autosomal dominant trait. So that's the only disease with dominant disorder. Okay, rest everything is yes. Rest everything is recessive. So those of you who gave me the answer for this question, kudos to you guys, because you have read through the question really well. Now this was the homework question that I gave in the previous class, and I saw a lot of you answering. I remember a few of your names, Mansi, Kumuda, Ayan. Okay, you gave me the answers. You did a really good job. And those of you who didn't write then, write now. A plant having the genotype capital A small a, capital B small b will produce dash kinds of gametes. Try it out. So Prakriti, Yashwant, Alan, Anand, Lakshmi, kudos my dear children. And Prakriti says myotonic dystrophy is autosomal dominant. Kudos. Super good. Super good. 14th A, 14th 4 is the answer. Okay. Give me in options A, B, C, D. What could be the answer? So there is a formula that you can apply to these type of questions. That is 2 raised to the power n. What do you mean by n? The number of heterozygotes. Ma'am, I don't understand what are heterozygotes. So over here, capital A, small a, they are hetero. This is one heterozygote. Capital B, small b, this is another heterozygote. Why it's heterozygote? If it would have been like this, it would be homozygous condition. Since one capital allele, one small allele is there, you call this heterozygous. So over here, 2 of them are there. So 2 raised to the power 2, it will be 4. You will find 4 different type of gametes. Please do not forget this formula. It can be coming. Okay, so now we have another person out there. Tejas, hi dear. Okay, so your answer for this question would be B, and who all got it correct? Prakriti, Alan, Anand, Tejas, Yashwant and Lakshmi. So all my dear children are doing super good. Next question. Question number 15. Which of the following is not a pair of opposing characteristics that Mendel studied? So this is regarding your monohybrid cross, where P plant was taken, and in P plants you have 7 characters, right? So you have to look for that. Ma'am, create polls. So we do not have we have not introduced the software for creating polls, but definitely doing it really soon. Okay, Anand? Evolution chapter would be coming towards the end, Santosh. I would be covering it. Tejas says the answer is D for this question. I see someone picking up. Okay, Prakriti also says it's D. Yashwant also says it's D. Okay, so I see 20 people watching this video and only few of them commenting, but I'm really happy for those who are practicing questions out here. Lakshmi 15D. Okay, everyone is saying D, which is why I'll lock this. Why this is the odd one out? Why this is not the pair? Because it was not pink and white color flowers. It was violet and white color flowers that was studied. Okay, it is not pink and white. So those who know the details know the answer as well. Axial and terminal is something that he studied. Which one is dominant over here? Axial or terminal? So axial flower means, suppose this is a branch. From a place a branch has to come, there a flower is coming. Terminal means over here at the terminal position, suppose a flower is coming. Which one do you think is dominant? Axial or terminal? Let's see. Let's see. This or this? Axial or terminal is dominant. This is out of the box question which just came into my head. Terminal. Abba. Yes, Lakshmi, he did. Mendel didn't take pink and white flowers. Instead, he took violet flowers. Okay. It's not terminal which is dominant. It is axial which is dominant. Children, axial is the dominant character. Remember, A comes before T. You can remember like that. So, A is dominant. Okay. Look, this is how questions can be coming around. Now, full and constricted pods. Which one is dominant? Full means a pod is like this. Okay, with green peas inside it. And constricted means it is very dry. It is like this. So, this is full and this is constricted. You also call the full as inflated. Which one is dominant? Full or constricted? Full, exactly. This is dominant. Now, green and yellow pods. So, when you go to market or when your father or mother goes to market, which one is the thing that they pick up? Green color pods or yellow color pods? Green is dominant or yellow is dominant? In the case of pods. In the case of pods, which is dominant? Green is dominant. Okay, green is dominant. Now, one last question to you guys. If you talk about seed of the green pea, green is dominant or yellow is dominant? Green is dominant or yellow is dominant in the case of seed. I am asking about seed. Yellow. Yellow is dominant. Even though we have green peas, but the yellow color is the dominant one. We saw that in the dihybrid cross experiment, right? Where round and yellow was fused. Remember? So it was yellow. So these type of questions you need to remember. That means you have to remember all the features. How many traits are there? There are 7 pairs of contrasting traits, and if you take in total, there are 14 contrasting traits out there. Okay, 14 in number in total. Okay, now going ahead. So 2 of them are related to seed color and seed shape. Then same thing goes for flower as well, flower position and flower color. Then pod shape and pod color. One unique thing is just stem height. Tall is the dominant trait. Okay. Now going ahead to the next question, try this question. So those of you saw this question, again as I always say, will get chocolate if you ever see me. Okay, I see lots of new people coming up, commenting down the answers. Ashwini, hi dear. And those of you who gave me the answers, Alan and Prakriti, Anand, Lakshmi, Yashwant, all of you doing really good, okay? Dhanush. Chalo. Next question. In the F2 generation of a Mendelian dihybrid cross, the number of phenotypes and the genotypes are. So, in dihybrid cross experiment, I just told you 9:3:3:1 is the ratio, where 9 of them are what? Round and yellow. 1 of them is what? Wrinkled and green. Now, you make new pairs, round and green and wrinkled and yellow. So, 4 phenotypes are there. Physical appearances, 4 are there. How many genotypes are there, my dear children? How many genotypes are there? 16. Hi, Dignath. 16A and 16D is what I see over here. Genotypes are actually 9. Prakriti is actually very correct. Kudos. Lakshmi is also correct. Ayyo, Wait. Yeah. Lakshmi is also correct. 16th answer is D. Listen, my dear children, number of genotypes is not 16. So what children usually do is 9 plus 3 plus 3 plus 1 is equals to 16 genotypes. Remember, certain genotypes are repeating again and again. So you just have 9 unique genotypes over here. So you have to remember like that. Genotypes are just 9. And how do you remember? Since you remember the phenotypic ratio 9:3:3:1, right? The first number you can remember, 9. So 9 genotypes are there and 4 phenotypes are there. Okay? Agreed? Done. Can we move ahead with the next question? Champs. Understanding, not understanding? Give me a thumbs up if at all you are understanding and if at all you want me to go ahead with the next question. Okay, Prakriti gave me thumbs up. What about others? Okay. Anand also gave me thumbs up, which is why I'm going ahead. 17th question. Mendel's work was rediscovered by 3 scientists in the year. So Mendel's work, no, Alan also gave me thumbs up. Thank you, Yashwant. Everyone are there. So Mendel's work, no, was rediscovered by 3 scientists in the year. What was the year, my dear children? When was it rediscovered? Any guesses? B is your answer, Yashwant. Lakshmi also says it's B. Ashwini also says it's B. Prakriti is asking me whether it's C. Is it C? Alan says it's B. Santosh is back saying it's B. Yes, it is B. 3 scientists' names were what? De Vries, Correns and Chemark, and they rediscovered Mendel's work in the year 1900. Please never forget it. Okay, 1900. This is very, very important question. Cool. Now going ahead with the next question, and before I go ahead with the next question and you answer this, can you answer this very important question? For how many years did Mendel did his experiment? So he started from 1856. When did he stop? For how many years did he do his experiment? You can let me know in the comment section of this video. Not here, in the comment section of this video. This is your homework number 1. Put homework number 1 and tell me the answer. Go through your NCERT and look, and then you write the answer for this. Okay? 7 years is what Prakriti says. Is it correct? Did he work for 7 years? Yashwant also says 7 years. So homework question is answered here itself. Yes, it was for 7 years, from 1856 to 1863. Remember the years, it can be coming in the question. 1863 for 7 years. Okay. Next, genes located very close to one another on the same chromosomes tends to be transmitted together and are called. Try this question. Genes which are there, which are very close to each other, they usually get transmitted together. What do you call these genes as? A, B, C, D. So Prakriti, Yashwant, Lakshmi, Dignath, all of you guys, my cute little champions are correct, okay? Now try this. Genes which are located very close to each other, what do you call them? C. Alan says it's C. Prakriti also says it's C. No, Yashwant says it's C, sorry. Ramesh, hi Ramesh. So I see new people out there. Kudos, keep going. Anand says it's C. Okay, all of you says it's C, which is why I lock C. It is known as linked genes. Remember linkage and recombination? Okay, the genes which are very close to each other, they are known as linked genes, as the name suggests. And they will be transmitting together. So the more they are close, the more they will be transmitted together. If they are very far from each other, they will show more amount of recombinations. So your answer is C. Next, chromosome number 19. Question number 19. Chromosome maps or genetic maps were first prepared by. Chromosome maps or genetic maps were first prepared by. Can you guess? Come on, my dear children. So, what do you mean by maps? So, over here, suppose I am able to understand that A is over here, B gene is over here. This is what you mean by genetic map or chromosome maps. Now, you can tell me the answer. So, Ramesh says the answer is A. Okay. What about others? What about others? And Prakriti says, what are allelomorphs? Allelomorphs means alleles, different alleles. For example, capital T, small t, they are allelomorphs. Okay? Anand says it's C, Lakshmi says it's D. So I am getting different A, B, C, D. You are giving me multiple choice questions, right? And Maria Babu, it says it's B. Okay, fine. So everyone is giving me MCQs, which is why I will give you the answer. And very good evening, Manasa dear. So the answer is D. Always remember, there was a teacher. His name was Morgan. Just like Jasmine ma'am, a teacher was there whose name was Morgan. And you students, my dear children, are what? Sturtevant. His student was there. So Morgan was the one who brought what? Linkage and recombination. He said that, okay, these genes, if at all they are this far, they will show this much amount of recombination and things like that. And depending on the percentage of recombination, his student was able to find the location of the genes. Okay, this is how, which is what you call as genetic maps. So what is your answer for this question? It is D. And now, if at all you're asked this question, who was the person who gave down linkage and recombination? It was T.H. Morgan, the teacher. Okay, now you never forget it. I hope you do not forget it. So ST for student, you can remember like that. Chromosome maps or genetic maps. Cool. Can we go ahead? Chalo, we will go ahead. And my dear children, are you liking this video? If at all you think this video is nice, I am able to explain you well, do not forget to hit the subscribe button right now. Share it with your friends who are in need, and also click on the like button. Yes? Chalo. 20th question. A couple has 3 daughters. What is the possibility of them having a son next time? What is the possibility of having a son next time? A couple who were there, they were having 3 daughters. So 20 C, and Yashwant also says it's C. Lakshmi gave me thumbs up, thank you dear. And Lakshmi also says it's C. So everyone, since they are saying C, can we log this option? Alan also, Yashwini also, everyone is C. So yes, the answer is C. Why, my dear children? Can you guess? Suppose there are 2 people out there, they have 3 daughters or 3 sons. Can you guess the other child, whoever is going to be born, can you guess their sex? No, obviously no. So always there is 50% chance for a male to be born and 50% chance for a female to be born. If at all the question was, a couple has 3 daughters, what is the possibility of them to have a daughter again? Would be again 50%. Okay, I hope you get it. So your answer is C, not B. So let me see who all got it correct, and by then you read this question and tell me the answer for this. So Prakriti got it correct, Yashwant got it correct, Lakshmi, Alan, Ashwini and Maria Babu, and Ramesh also got it correct. Dhanush, Baba, let's check Marty once more. Okay, I think we are on different question, or the question number has been mismatched. 21, Prakriti is on fire today, says it's D. What about other people? Let's show that everyone is equally on fire. Manasa is also saying, Manasa is back in action and says 21D is the correct answer. So when is it possible for a woman to be colorblind? Listen, color blindness is a sex-linked recessive disorder. That means for a male, it can be this condition where he gets the color blindness. Only one X chromosome is there is no other X chromosome, he can get the disease. But for a female, both the chromosomes has to be affected. Both the chromosomes has to be affected, has to be recessive condition. Then only the person would be having it. So when do you think that a woman can be colorblind? When she gets one chromosome from her father. So it has to be like this, and when she gets at least one chromosome from her mother. So her mother can have a condition like this, or she can be like this as well. Okay, she can be either colorblind or she can be carrier. Now over here, you guys are saying the answer is D. Let me check. The father is colorblind and the mother is a carrier. Okay. The father is colorblind and the mother is has a normal vision. So this is also normal vision, but as you said, you guys are absolutely correct. You have to mention that she is a carrier. Normal vision can be just like this also. So this is also normal vision, this is also normal vision. But this is additionally, they are carriers also. So you have to say the answer is D. So those of you who gave me the correct answer, kudos. Okay. Let's go ahead with the question number 2. So 22. A man marries a woman. Both do not show any apparent traits of inherited disease. 5 sons and 2 daughters are born, and 3 of their sons suffer from the disease. However, none of the daughters are affected. The following mode of inheritance for the disease is. Ma'am, even when both the parents have the genes, there are still 50% of the daughter to be affected, right? Like she can be affected, or when both the parents have the gene. Listen, if both the parents have the gene, if the mother is a carrier, if mother is just a carrier, then there are chances that the female can be a carrier. Otherwise, if the mother is not a carrier, she has color blindness, and the father is anyway colorblind, then in that condition all the progenies would also be colorblind. So mother has to be a carrier if the daughter has to be a carrier. Okay. Now over here, you guys have started giving me the answer. Let me read out the other questions answer. Prakriti gave me the correct answer, Manasa gave me the correct answer, and Yashwant did, Anand did, Ellen did, Lakshmi did, Anne Maria did. All of you doing really, really great. Now we are solving this question, 22nd question, and I see Alan saying it's B. Lakshmi says it's D, A, B. So you guys are giving me an option that I have to tell the answer, right? Prakriti, I will be back to your question, okay? Just give me a second. So my dear children, over here, let's decode. A man marries a woman and both do not show any apparent traits of inherited disease. Both are not showing any trait. But whenever the question comes like this, you have to think for the women once. Because women, my dear children, can be carriers. Okay, can be carriers. So have that option that this particular lady can be a carrier. Now, 5 sons and 2 daughters are born. 3 of their sons suffer from the disease. See, I told you, since she was a carrier, my dear children, the sons which were there, which were produced, they had the disease. How many of them? 3 of them. And the daughters were not affected. The following mode of inheritance for the disease is what? So you get to understand the female is having the disease and she is passing on to the sons, and none of the daughters are affected. That means it is what? X-linked recessive disorder. It is a recessive disorder. It cannot be dominant. If it was dominant, the women might have had that. She cannot be a carrier. If at all one gene is affected, that means she would be having that disease. But here there is no trait that is being seen. Okay, and it is not autosomal because over here it is clearly seen that sons are more affected. And if it was autosomal, both of them would be equally affected. Which is why your answer would be A. Okay, it's okay, Alan, no problem. Your answer is A. Now coming to Prakriti's question, ma'am, I meant when the mother is carrier and the father is affected, it will be still 50% right. Lakshmi says she's confused. Okay, let's go one by one. This question, let's solve it once more. Here, the man marries a woman and there are 5 sons being born, right? Out of the 5 sons, 3 of them are affected. 2 daughters are also there, but they are not at all affected. With that, we get a clear idea that it is not clearly autosomal, because if it was autosomal, autosomes are there in everyone, male and female, so it does not matter. It can be either of them being affected. It will be equal affectance of both of them, right? But here clearly the males are affected. That means you're getting an idea that it is X-linked or sex-linked, my dear children. Now whether it's recessive or dominant, that is the next question. So if it was dominant, okay, if it was dominant, since she has one particular chromosome being affected, this particular woman would have definitely shown the disease over here if at all she was dominant. But it is clearly mentioned that both of them do not show any apparent traits of inherited traits, disease out there. That is why we say it's not dominant disorder, it was what? Recessive disorder. Dominant, you cannot show that. Is that clear, Lakshmi, now? Is it clear somehow? Read the question once more. It will be all the more clearer to you. Okay. Now solving Prakriti's question. Ma'am, I meant, so mother is a carrier, suppose. And you want what? Father is affected. The father is affected. In that particular condition, my dear child, you have XC and Y over here. And here, this and this comes together, you get XC, XC, that means the female is affected. This and this comes together, that means female is a carrier. This and this, XY, male is normal. This and this, XCY, male is affected. So out of, all of them, 25% of the male is affected, 25% of the female is affected, 25% of the female is carrier, and 25% of the male is going to be normal. Ayo, going to be normal. Prakriti, did I solve your doubt? If you're just talking about female daughters, out of the daughters, yes, 50% of them would be affected and 50% of them would be carriers. But if you're talking about whole, then you have to take 25%. Okay? Going ahead with the next question out there, which is this. Okay, you meant only the daughters, then you are correct. Yeah. Next question. If both parents are unaffected carriers of thalassemia, try solving this question, okay? If both parents are unaffected carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in affected child if both of them are unaffected carriers? Remember, thalassemia is an autosomal recessive disorder. It's autosomal. 23D, sorry, 23D, Lakshmi says it's A, Alan says it's A, Yashwant also says it's C. Okay, so D, A, C is what I get over here. Listen, it's an autosomal disorder, right? So both of them can be carriers of this. Over here, both of them are carriers, small r is affected. It's a recessive disorder, that means the small r, when it combines together, that only someone can get the disease, right? So suppose it crosses like this. This and this crosses, you get capital R. This and this goes, you get capital R, small r. This is not affected. Again, capital R, small r, only 25 out of 4. Okay, out of 100, only 1 by 4, my dear children. So basically 25% of them are going to get the, what do you call, the disease out there. Okay, 25% of them are going to get disease. So your answer would be A. Yes, Manvita Reddy, hi dear. Fine. Let's go ahead with the next question. Dear children, stop me if at all you have any doubt, okay? So those of you who said D or C, my dear children, I hope you have corrected that mistake. It would be A, okay? Since it is an autosomal recessive disorder. Praveen is also there. Hi Praveen. Try to solve this question. Question number 24. Come on. Now, when you try to solve question, can you please give me the answer like 24 A, B, C, D so that I have an understanding of what question are you solving? So, the fact that every gamete formed has either of the alleles for a trait follows what? So, for example, capital T, small t is that of the parent. Now, it is saying that every gamete formed by this parent would be having any one of these alleles, either capital T or small t. What law does it show? What law does it show? A, B, C, D. Sometimes the questions can be framed differently. So everyone is saying 24D, kudos to all of you because you all are wrong. Think, can it be 24D? Fact that every gamete formed has either of the alleles for a trait. You're just talking about one trait, not multiple traits. Always remember this very important thing. If it is one trait, you're talking about law of segregation. Law of segregation says that alleles of a trait separate, okay, and go into the gametes. So this is law of segregation. It is not D. Law of independent assortment is different. Law of independent assortment is when, for example, you have capital R, small r, capital Y, small y, dihybrid cross experiment, right? Over here, the chromosomes are like this, capital R, small r, capital Y, small y. Now, when you form the gametes, from here capital R can go, from here capital Y can go. Okay, now again, from here capital R can go, from here small y can go. So they are assorting independently. Capital R does not say I will only go with capital Y. So the alleles for one trait and alleles for another trait are assorting independently. That is law of independent assortment. You see that in dihybrid cross. Here only one trait is being asked, so you have to look for this particular thing, law of segregation. Okay, I hope you have got it. So those of you who said the answer is, D, please make sure you're going through it once more. The answer is C. Manasalayo. Any Malayalis up over here? Okay, chalo. Next, we would be going ahead with the next question out there. It's from Human Health and Disease. But before I go ahead, my dear children, are you understanding all the things? If yes, give me a thumbs up. Can we move ahead to the next chapter, that is Human Health and Diseases? Or do you have a doubt? Please solve it now. Tomorrow, when you have an exam, you cannot go there and ask doubts, right? Anand says thumbs up. Anand gave me thumbs up. Thanks a lot, love. Thank you so much. Alan also gave me thumbs up. Okay. Now, Anne Maria, okay. Chalo, let's go ahead. 25th question. Listen, my dear children, we have 40 questions. Okay, we are on 25th question. We have to do it fast and we have to do it correct. Okay, chalo. My exam is day after tomorrow. Yay, we will rock the examination. Okay, you have extra time. So a chemical structure is given below. Identify its plant source. First. What is the structure of Atropa belladonna, Cannabis sativa, Papaver somniferum, Erythroxylum coca? 25B, 25C, B, C. Okay, tell me the answer. A, B, C, D. After this, what is the correct answer? B. B, yeah. Those of you saying B. No chocolates for you. Okay, okay. Now you're guessing. My dear children, listen. This is very important. It's there in your NCERT. It's not out of syllabus. Okay, and I'll tell you a way you can remember it. It looks like an O, right? O means opioid. We are talking about opioid. And where do you get opioids from? From opium poppy. And what is the other name for opium poppy? It is known as Papaver somniferum. This structure is of what? Is of morphine. Don't forget. Morphine is a drug that usually doctors give to what? To the patients who have underwent surgery for making down their pain, for making their pain go down. Okay, it's a pain relief and a sedative. Okay, so answer is C, Papaver somniferum. It's morphine. It comes under opioids. So O looks like this. And now you are giving me correct answer. Yes, painkillers. Now the same structure is also, one more structure is given in your NCERT, that is of cannabis sativa, okay, cannabinoid, where you can see a line like this. They have this line like this at the end in the chemical structure. So that is cannabis sativa. So you can see, think like C, C, C. You can, C stands for cannabis sativa. You can think like that. Okay. Now going ahead with the next question, 26th. You read this question, try it out, and by then I'll read out the name of the people who gave me the correct answer. So C was, Lakshmi gave me the correct answer initially itself. So kudos to you. And then I got the answer. Okay, then she's changed the answer. Okay, anyways. Anne Maria and Anand and Praveen. Anne Maria, yes, it's a painkiller. And Dhanush, kudos. Supriya ma'am, tomorrow is English exam. Can you teach English as no one? There will be a video released for English as well. Please don't be worried. Okay? Soon. Okay, 26. Okay, Yogaraj says is D, Lakshmi says is D, Dhanush also D, Prakriti also D. Let's see if at all D is correct or not. So one of the following is not the causal organism for ringworm disease. So ringworm disease is caused by 3 different type of fungi, microsporum, trichophyton, epidermophyton, but not macrosporum. So this is the odd one out and you guys are correct. So how do you remember it? Microsporum, little small spore. Okay, trichophyton and epidermophyton are basically fighting on our epidermis. You can remember like that, which is why we have skin, rashes. It is like ringworm. It forms like a ring-like structure. It's not formed by worm, it's a fungus. So trichophyton, epidermophyton fighting on us and they are small, microsporum, but not macrosporum. Okay, so your answer is D. Going ahead with the next question. 27th question, try to solve it out and by then I'll read out the name of the people who gave the answer. Lakshmi, Yogaraj, Dhanush, Prakriti, Praveen, Taj, hi, Anand and Anne Maria, Babu, you all are correct. Children, I hope you are taking something up from this particular class and that is knowledge. Okay? Please make sure if at all you're stuck at question, write the question number down, come and resolve the question, okay? 27th answer. Okay, 27th, it is B is what you say. Okay, wait. Intestinal parasite which causes blockage of the intestinal passage and whose eggs are excreted along with the feces of the infected person. That means along with the stool of the infected person, the eggs of this particular parasite is gone. What is this particular parasite? 27 B. I have seen only one option saying it's D. Well, the answer is not D. It's B and it is Ascaris. Okay, why is that so? Okay, it is Ascaris. So the name of the the parasite is Ascaris lumbricoides and it causes a disease that is known as ascariasis or also known as roundworm disease. It causes intestinal passage blockage. It causes bleeding also in some cases. So you all are correct. With B, you are doing kudos. Now 28th question. Solve it out and I'll read out the name of the children. Which of the following is affected by the infection of Wuchereria bancrofti? Wuchereria bancrofti is also known as filarial worm and it causes filariasis or elephantiasis. Now I've given you a clue. Tell me the answers. 28th, write like this, 28 A, B, C, D, so that I get to know what you're saying is correct or not. Abhishek says it's B, Anand says it's A, Komal Kaushik, you do have English, you would be having a video, recorded video for English, okay? And 28 A, okay. So all of you are saying A and your answer is correct. Why it is A, my dear children? So Wuchereria bancrofti, goes and affects the lymphatic vessels. And you know what happens to the lymphatic vessels? Usually what the lymphatic vessels do, they take in the extra fluid between the cells and they give it back to the blood. Okay, so that the extra fluid is not being lost. They give it back to the blood. So if at all the fluid is not being recollected, what will happen? There will be swelling caused out there, which is why this disease is also known as elephantiasis, because usually the limbs, the lower limbs are the ones that is affected and it is swollen. It looks like elephant's leg. Okay, so always remember it. Yeah, they become unusually large. So it is lymphatic vessels and mostly the, there would be gross deformities in the sexual reproductive parts as well. Okay. Now, going ahead with the next question, 28, 29, solve it up. By then, I'll read out the names of the children. This has been a wonderful task to do so because I am really, really proud whenever I read out. So, Anand, Abhishek, dear, I hope you have got your answer correct. Ramesh, kudos. Manasa, kudos. Anne-Maria Babu, awesome. Srinidhi, awesome. Abhishek, yeah, awesome. Manvita Reddy, Dhanush, all of you. Praveen Taj, Preetam, hi dear. And Prakriti. Okay, now we are solving 29th question. Try to solve it. 29th, which of the following pairs correctly matches the disease and the pathogen causing it? Correctly matches the disease and the pathogen that is causing it. Can you guess? 29 A. Typhoid is what you say? Yes, it is caused by Salmonella typhi. Typhoid is caused by Salmonella typhi and pneumonia is not caused by Haemophilus pneumoniae. It is caused by Haemophilus influenzae. Malaria is caused by what? Malaria is caused by, any guesses? Chalo, you tell me the answer for this also. Malaria is caused by what? And ringworm disease we already discussed is caused by Microsporum, Epidermophyton and Trichophyton. Any guesses? How is malaria caused? Who causes malaria? Navneet, thank you so much for this. Thank you so much for coming over here for the live. Hi. Plasmodium vivax. Yes, Anand. And yes, Preetam. Awesome. Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae. Okay. So in this, what is the most malignant form of Plasmodium? Any guesses? Streptococcus. No, Baba. Streptococcus is for, yeah, I think you have given the answer for pneumonia, right? Which one is the most, malignant form of Plasmodium which causes malaria? P. falciparum, exactly. P. falciparum is the one that causes the malignant form of malaria. Do not forget it. So P. vivax is there, please. P. malaria is there, and P. falciparum is there. Out of that, P. falciparum is the one that causes it. Okay. Next, hemozoin is a. What is hemozoin, my dear children? What is hemozoin? So, my dear children, do you know that you guys are the best children out there? Because you all are attempting the answer, however wrong or right or whatever is there, you are giving your attempt and that means a lot. It's a toxic substance. 30D is what is someone saying. Who is saying that? Preetam is saying it's 30D, someone is saying 30C. What is the answer for this? So 30D is toxin released from Haemophilus infected cells. No, it is not. Haemophilus influenzae is for pneumonia. Hemozoin is released in a person who is infected from malaria. What exactly happens over here? So malaria is caused by Plasmodium. So your answer for this question is toxin released from Plasmodium infected cells. So listen to me carefully. This is a story. Okay, this is a story and this will help you in your examination. So Plasmodium has come to the RBC thinking that it will get the food. So it is going to break down the RBC. In the RBC, what is there? Hemoglobin. So hemoglobin is the food of this particular Plasmodium. So Plasmodium is like, I just need the food, okay? But then, when it breaks hemoglobin, hemoglobin is broken down to two things. One is heme and the second thing is globin. You know what? This heme is very toxic for the Plasmodium. So the Plasmodium immediately converts this heme into what? Hemozoin. It converts the heme into hemozoin. Now everything is happening inside the RBC, and not just that, they are doing this and they are reproducing as well. Okay, I mean they are undergoing asexual method of reproduction, not sexual, asexual method of reproduction, and they are increasing in their number. So once they increase the number too much, the RBC bursts open. But what's the sad part of the story? The hemozoin which is there, it is being released, and this hemozoin is toxic for us. It is not toxic for them, it is toxic for us, which is why we get fevers and chills. Okay, I hope you never forget this answer now. The answer is C. Understood? Give me a thumbs up if you understood. So 30, let's start from, okay. So Anand, Dhanush, we have to check the answer, okay. So Anand, Lakshmi, Prakriti, Alan, Navneet and Abhishek and Maria. Everyone have given me the answer. Ramesh also gave me the answer. Kudos. Chalo. And I am saying children are giving wrong answers and then they correcting it as well. Good, 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 good. Keep going. Superb. Now, over here there is a picture of Plasmodium's life cycle. Do you want me to explain this now or towards the end of the session? Because this is very important. On the basis of this, you are going to see few more questions around there. You want to explain now? Then tell me now, or if you want me to explain later, you tell me later. End. Chalo, okay. I'll do it at the end. That means you are going to solve it, the questions by yourself, right? So yes, start solving. During the life cycle of Plasmodium, gametocytes develop in. During the life cycle of Plasmodium, gametocytes develop in what? Okay, okay. Now questions. Okay, we will do questions now. Later we will explain. 31D. During the life cycles of Plasmodium, gametocytes develop in. That means your question is, where are gametocytes being formed? Where are gametocytes being formed? So I see 31D is answer, 31C is Manasa says it's A, Dhanush says it's A, Alan says it's A, Abhishek A, Navneet A, Lakshmi C, Navneet A. Okay. So the answer for this question is A. It is not C, it is not B, not D, nothing else. Which is why I told you, do you need an explanation for that? So listen to me. In the case of Plasmodium, they enter in sporozoite stage. They have different stages in our body. Once they reach RBC, once they reach RBC, they are going to transform into a stage that is known as gametocyte. Gametocyte. So it is in the RBC where gametocyte stage is being formed. The next step of them would be to produce the gametes, but that does not happen here in our body. Now, the gametocyte is there in the blood. A mosquito comes and sucks the blood. The gametocyte gets into the gut of the mosquito. Inside the gut of the mosquito, these gametocytes will give rise to gametes and they will fuse there. So sexual reproduction, always remember, sexual reproduction happens in the body of the mosquito. Asexual reproduction happens over here in us. Okay? And now you're saying no. So your answer for this question would be A. You want the explanation now, it seems. So listen to me carefully. I'll do it really fast. You don't have to be worried. Okay? So listen, this is Plasmodium. It causes malaria. It is a protozoan. I hope you know that. So now Anopheles mosquito is there. A mosquito that causes malaria, right? In the Anopheles mosquito salivary gland, in their saliva, they have sporozoite stage of this particular organism, that is Plasmodium. So mosquito comes, bites us. When it bites us, it does not give us the blood. It basically puts in saliva, and so the saliva has some clotting, anti-clotting enzymes, so that the blood can easily come around. So through the saliva, what is there in the saliva? Sporozoite. So sporozoite gets into the human body. Now in the human body, the terrorist which is there, okay, they take the first camp, which is the liver. So terrorist first camp is liver. Remember, the cells of the liver are known as hepatocytes. In the hepatocytes, they will undergo asexual reproduction. Once it is done, they will get lysed. The hepatocytes breaks open and the asexual mode of whatever product is gotten, it goes into the blood. Now in the blood, these cells, my dear children, target the next thing, that is RBC. In RBC, again they will undergo asexual reproduction. And what will happen? The RBC will get lyased. When the RBC get lyased, I already told you what will happen. Hemozoin which was there, that will be released. Apart from that, gametocytes are going to be released. Now everything is there in the blood. Gametocytes are not forming gametes, they're just there in the blood. Mosquito comes, takes this blood, and now the gametocyte is in the body of the mosquito, right? It goes into the mosquito's gut. Gametocytes will give rise to gametes in the mosquito's guts, and these gametes will be fusing and they will be forming something. Later again there will be transformation of phases, and again it forms sporozoite phase which goes into the salivary gland. So over here in the salivary gland at the end you will find sporozoite stage of Anopheles mosquito. Okay. Yes, that small thing has salivary gland. Ramesh, I was saying earlier also. So Ramesh is the most, what do you call, clever boy out here. Okay. So Anand, ma'am, Kannadallu swalpa heli. Okay, my Kannada is not that great, but I am definitely trying to learn it. I hope I have explained you in, this is not human liver, okay, it's RBC. Okay. Children, did you understand this thing, the life cycle of Plasmodium? It's okay? If it's okay, give me a thumbs up. If it's not okay, don't give me thumbs up. Give thumbs down. I don't know what to say. Okay? Shall we move ahead to the next question? 32nd question. So it's very important, the life cycle. Always remember the stage in which we are infected is sporozoite stage. The stage in which it goes out is gametocyte stage. No sexual reproduction is happening in our body, and the toxin that is released is hemozoin. Sexual reproduction happens in the case of mosquito's body. Okay, you all gave me thumbs up. Thank you so much, dear love, my dear children. Okay, now over here, 32nd question. The human immunodeficiency virus primarily targets and destroys what? 32B, Abhishek says 32B, and Dhanush says 32B, Alan says B, Anne Maria says B, Manasa says B. Where is Prakriti? Prakriti is missing, Navneet is missing. Okay, Prakriti is back. 32B, okay. So the human immunodeficiency virus attacks the T helper lymphocytes. You guys are correct. If at all it is asked, what is the factory of HIV? It is macrophages. So usually HIV attacks the macrophages. There they increase the number, and then they go to T helper lymphocytes and try to damage them. Because you know what? These guys are the best guys of our body. They help every cells out there in their, what do you call, mode of action. And if at all this person is not there, the mediator is not there, do you think anything will happen? No. So which is why they target the most important thing, that is the T helper lymphocytes. I am making coffee, that's why. Okay. Okay, so your answer is B. Hi Navishri. So all of you who got the answer B, you are absolutely correct. Now going ahead to the next question. The substance produced by a cell in the viral infection that can protect other cells from further infection is what? The substance that is produced by a cell and that can protect other cells from further infection is what? 33C is what Anand says. Okay. So over here, suppose a cell is infected by virus. So when the cell is infected by virus, my dear children, it releases some things such as interferons. Why? Because these interferons will be telling the other cells to be safe, that there is an attack from the viruses. So these are known as cytokines, the things that are released by the cells which helps the other cells to be aware that something is there. It protects them. So the answer for this question is interferon. Okay. So who all gave me the correct answer? Let's see that. And before, okay, I'll go to the next question. You try this and I'll read out. So C30, Anand says the correct answer. And Prakriti also gave me the correct answer. Preetham gave the correct answer. Dear children, it's not D, okay? It's, as I told, it's not D, it's C, interferon. Viral infection, interferon. Now 34B, try it out. Lakshmi is back. 34B is what she says. Antibodies present in the colostrum which protects the newborn from certain diseases is. Which protects the newborn from certain diseases is IgE. No. Your answer for this question is IgA. Listen, newborn. A for Amma. A for Amma. So Amma gives what? The first milk which is there, which is known as colostrum. And colostrum has high amount of IgA. You can remember like that. Okay. So the answer is IgA, the option is B. Okay, if at all you are asked, what is the first antibody that is passed from the mother to the child? What is it? What is the first antibody that is passed from the mother to the child? What is it? I'm not talking about colostrum. First antibody passed from the mother to the child. Try it out. And those of you who gave me correct answer 34B, let me read out your names. Lakshmi, Anand, Prakriti, and Maria Babu, and Manasa, and Dhanush, and Alan, Ramesh, as well as Lakshmi. Yes. IgG, IgE, everyone is giving me other answer. Okay. So listen, the first one, you take it in this flow. These many antibodies are there, gamed. Okay. The first antibody to pass from the mother to the baby is this, that is IgG, and this is passed to the placenta. Then when a mother actually becomes, what do you call, after the delivery, okay, after delivery, with the help of colostrum, the first milk, she passes IgA. M and D is not given in your NCERT as in what is their function, but you can just remember IgE has high resemblance to allergy, so it is found in allergies. IgE is there in allergy. Okay, you can remember like that. This trick would definitely help you out. Okay, so it's IgG, and I see Anand was the first person to give me the correct answer, and then Anne Maria, and then Abhishek, Mansa. It's okay, dear. IgE causes allergy. Yeah, IgE is the one that increases their amount during an allergic reaction. Okay. It's not like it causes allergy. Yeah, it basically technically causes allergy as well. Ramesh Singh, what's your mother tongue? My mother tongue is actually Malayalam, but I was born and raised up in Madhya Pradesh, Indore. So I know Hindi, Malayalam as well as English. Okay. Okay. Vaccines work by what? 35A, B, C, D. 35A is what Dhanush says. Alan says A. Okay. Ma'am, then is histamine one of Ig? I'll tell you, Prakriti, right now. Okay, there's a question coming around it. So, you guys are saying A, vaccines work by stimulating the production of antibodies. The answer is correct. So basically what is happening, vaccine is a weakened form of pathogen. Okay, we are giving a weak form of pathogen into the body, a weakened or some neutral form of the pathogen we are giving to the body, such that the body has an understanding that okay, this type of things can come in and you have to produce antibodies later on. So the first time we get a vaccine, our body reacts to it by production of little amount of antibodies and memory cells. But this response is slow. This is known as primary response. But the second time anything comes up to us, my dear children, they will produce it more amount. So for stimulating the production of antibodies, we have vaccines. Okay. Now can you tell me hepatitis B vaccine? There's a question. Hepatitis B vaccine was produced with the help of which organism? Which organism in recombinant DNA technology? Hepatitis B vaccine was produced by the help of which organism in recombinant DNA technology? Can you try to tell me the answer for this question? This is given in your NCERT below the paragraph of vaccine. There's one line given and with that you can tell the answer for this. Rata, no. Yeast, exactly. It was yeast. Hepatitis B vaccine was produced with the help of yeast. That is also there in your human health and diseases chapter. So please go through it. Okay. Yes, Dhanush. Yes, Lakshmi. All of you right. Okay, let's go ahead with the next question. Yes, which of the following cells actively participate during allergy? Now you can tell me. Which of the following cells actively participate during allergy? And over here, I see all of you giving me the answer, starting from Dhanush, Alan, Ramesh, and then Lakshmi, Preetham, Prakriti, Anne Maria, Manasa, all of you doing really, really great, okay? Easter day, yes, you can remember like that. So Anand says the answer for this question is C, mast cells. Okay. Dhanush also, Lakshmi also. Okay. So over here, yes, the answer for this question is what? Mast cells. Which of the following cells actively participate during allergy? So listen, over here, there is mast cells, and on the surface of the mast cells, you have IgE antibodies. Okay, now the IgE antibodies reacts with the antigens out there, and the mast cells, on the reaction of these antigens with the IgE antibodies which are present on the mast cells, the mast cell is going to release histamine. It is going to release histamine as well as serotonin. Histamine as well as serotonin. And you know what do they do? The blood vessels which are like this, which are narrow, they will dilate it. They will dilate it. Why? They want all the other immunological cells to come at this particular place, which is why, my dear children, we have swelling and everything out there. So this is it's an exaggerated reaction that is happening because of the production of histamines and serotonin. Always remember, whenever we have allergy, doctors gives us antihistamine. If at all you have heard of this, medicine out there, which is known as cetirizine. It is an antihistamine drug. Okay. And also when you go to the doctor and if he wants to check if at all you have an allergic reaction, they check for IgE. If at all the amount of IgE is elevated, that means you might have an allergic reaction. So all of you might have got a feel of becoming a doctor as of now, and I'll go ahead with the next question. That is this. Okay, try solving this. This is of evolution chapter, and I will read out your answers. So those of you who got the correct answer is Anand and Dhanush and Lakshmi and Preetam. Rest all of you gave the wrong answers, but it's still fine. It's still okay because you guys tried. I love when you guys tried. Okay. When you do not try, when you just sit and watch the video and you think that you will remember it, that's the worst way of dealing with a live class. You should participate. 37B is what Preetam says. Okay, let's see. So match the type of man with their origin periods and choose the correct answer. This is column 1 and that is column 2. Ramapithecus, when were they found? Australopithecus, Homo erectus and Neanderthal man. I'll tell you a story. Okay, not a story actually, a way to remember it. So first we have Dryopithecus and Ramapithecus. Ramapithecus has Rama name in it, so you can remember like that. Ram, human-like, right? So Ramapithecus were more human-like. They were there found 15 million years ago. Ramapithecus and Dryopithecus 15 million years ago. So this goes with, A goes with 2. A goes with 2, I just have one option, which is why this becomes my answer. Okay, which is why this becomes my answer, but I'll explain it to you. So Rama guy came in, okay, over here 15 million years ago, and after that, but all of them, my dear children, Ramapithecus and Dryopithecus, they used both of their limbs for walking. They used both of the limbs for walking, but were still man-like. Then later on, next thing came up, that is Australopithecus. Almost 2 million years ago, so after 15 million years ago, 2 million years ago, my dear children, these guys came in, Australopithecus. Okay, they finally started to stay like this. They used to walk on 2 limbs out there. Okay, they didn't use their 4 limbs. They just used their hind limbs, Australopithecus. Now, came the Habibis out there. After Australopithecus came the Homo habilis, or you call it Habibi. So they also came around that time, and they didn't used to touch meat. They were vegetarians. They used to make tools and everything. Now, finally, okay, after the Homo habilis came Homo erectus, who were totally standing erect, my dear children, and they were the the first human beings to basically start eating meat and all. Okay, they started eating meat until now. Until Homo habilis, they were eating fruits and vegetables and things like that. Homo erectus, they started eating meat. They used their whatever tools were there to kill the other organisms and eat it. This happened. Homo erectus basically came around 1.5 million years ago. Okay, and from Homo erectus you have two organisms. One is Neanderthal man and the other one is Homo sapiens. First came Neanderthal man almost 1 lakh to 40,000 years ago. They were the first people who were emotionally connected. Okay, they started burying the dead, their relatives out there, and they started covering their body as well. That was Neanderthal man. And towards a little almost 75,000 years ago, my dear children, came Homo sapiens, that is us. Okay, we also came into the picture. After that was the last thing, that the modern man is the Homo sapiens out there. So this is the story of everything. I hope you will remember it in some ways, if at all you can. Now we are developing from Homo erectus back to Ramapithecus. I think so, yes. So your answer is B. Please remember, it's not just this that you have to remember, but the flow of the things as well. So today in a session, I said a statement, I do not remember it now, but if at all I find the statement, I'll put it. Okay. Anyways, 38th question. Evolutionary convergence is characterized by. Evolutionary convergence is characterized by. So all of you who gave me the correct answer, let me see. Preetham, Alan, and Anne Maria, Dhanush, as well as Anand gave me the correct answer. And now you guys have to try for the next question. Evolutionary convergence is that means convergence. Okay, what do you mean by that? Convergence means separate things converging to a same point. That is what you mean by convergence. Lakshmi says the answer for this question is B. Development of common set of characteristics in a group of different ancestry. Yes, your answer is absolutely correct. So different ancestors were there, but now they are developing what? Common set of characteristics. Converging to a point. Simple. You just have to use what? Common sense over here. So, if development of dissimilar characteristics in close related groups, that means they are moving away. No, we don't want. Development of characteristics by random mating, no. Replacement of common characteristics, no. Same. The only answer for this question would be B. Okay. Can we go ahead? The 39th question out there. Let's go. Replacement of lighter colored variety of peppered moth, that is Biston betuluria, to its darker variety, that is Biston carbonaria, in England is the example of what? Is the example of what? 39th question, my dear children. 39th A is said by Preetham. Darshan is there. Hi Darshan. Says it's B. Okay, I do not know which question, but 39, you're saying it's B. Okay. Prakriti says it's A. Anne Maria says it's A. Alan also says it's A. Monisha is there. Hi. Dhanusha. Everyone is saying it's A. So yes, the answer is A. Listen. Initially, there was the lighter variety of moth. Its name was what? Biston betuluria. Okay. And then came the darker variety. So over here, the lighter variety of moth, they were able to survive because the plants were having something on the top of it which is known as lichen. Okay, lichen. So which is why even the lighter variety of the moth, they were able to survive. And over here in this light background, if you put something black, don't you think they would be caught? They were actually caught. So the darker ones, they died off, and the lighter ones said that, see, you cannot survive anyways. But then after years, because of humans' anthropogenic activities, that means human beings' activities of pollution and everything, my dear children, the lichen which was there, it was not able to survive. It cannot survive in polluted areas, which is why every tree bark which was there, it turned to be black in color. Now in this black background, the black ones can definitely survive, but do you think the white one can survive? The white one cannot be survived because it can be clearly visible out there. So this time the white one died, or the white one were replaced. Okay, so this is known as industrial melanism. Because of industrial pollution, and my dear children, a lot of things happened, the color changes and things like that. And this was the nature who selected them. So initially the nature selected the white ones, and then the nature selected the black ones. They were able to adapt better. The few ones which were there, they were able to adapt better, and they increased the number because of something that is known as natural selection. So your answer for this question would be A. Okay? Okay. Can we move ahead? 40th question. Following diagram provides an example of what? So this is an picture that is given over here. There is Australian marsupials, and from here you have different organisms coming up. So what is this an example of? By then I'll read out the name of the children who gave me the answers. Let me see what is the correct answer. Yeah. So 39A, Prakriti gave me the correct answer, Preetham gave me the correct answer, and Anne Maria, Alan, Monisha, Dhanush, Lakshmi, Santosh, industrial melanism, dear. Okay. And yes, kudos to all of you. And now we are in 40th question, and all of you are saying D, starting with Preetham. Okay, following diagram shows divergent evolution. Do you think so? Well, the answer is yes. So this is an example of adaptive radiation as well. Remember, this is an example of adaptive radiation as well, where there is a common ancestor over here, and this common ancestor basically is moving to different areas and forming different organisms. So they have similarity, but now since they are adapted to different regions, they are showing divergent evolution. They are moving away. Okay. Now, if at all, so your answer is correct. it is what? Your answer is correct. It is divergent evolution. But if at all, I give you one more very important thing. You have to tell me the answer for this. So those of you tell the answer for this, again gets a chocolate whenever you meet me. That is, Australian marsupials are over here. Australian marsupials are over here. Similarly, placental mammals are also over here. Placental mammals are also from here. So from A team, A1 went there. From B team, B1 also went over here. From A team, A2 went over here. From B team, B2 went over here. What are they showing? What type of evolution do they show? A1 and B1 and A2 and B2. What type of evolution do they show? Divergent, convergent, or any other evolution do you have in mind? This can be a question. So you know, in the paragraph of Australian marsupials as well as placental mammals, my dear children, you are saying that basically all of them are, for example, from here if flying phalanger is going, from the placental mammals team also someone is going there. Ishan, yes my dear child, a video would be coming for English as well, is what I believe, yes. Parallel. No, it is convergent evolution. Why? Why it is convergent evolution? Because both of them, they are different. They do not have common ancestors, but they are coming over here, converging to a point. And since they are in the same adaptation, they will be having some features which are going to be similar, even though structurally, internally, they are not similar. But because they are getting into an adaptation, a geographical area, in the same geographical area, they will have some common, things out there, which is why it is an example of convergent evolution. So always remember, if only one thing is given, it is divergent evolution. If two things are given together, Australian marsupials as well as placental mammals, it is convergent evolution. Always remember that. Okay? I hope this is clear to you guys. They all are converging to a point. That is what you have to remember. If I had the picture of, that particular thing, I would have definitely shown you. Ma'am, is question and answer is going to be included in this video? No dear, I have not included that over here, since I thought it would be too much for you guys. Alan, understood. Thank you. Okay. Now, at a particular locus, by the way, this is the last question. After that, I would be taking up doubts. If you have any doubts, you can ask me. Tomorrow, I will be having a full-proof lecture, because I'm assuming most of the children have their exams the day after tomorrow, which is why tomorrow there will be a live regarding everything possible out there for all the chapters together. Okay, it's at 4 o'clock. Be there, be sharp there and participate. So last question, at a particular locus, the frequency of allele A is 0.6 and that of allele B is what? Allele capital A is 0.6 and small a is 0.4. What would be the frequency of heterozygotes in a random mating population at equilibrium? What would be the frequency of heterozygotes? So how do you calculate heterozygotes, my dear children? Basically, homozygote, for example, you have to calculate capital A. For that, you do P square. What is P over here? P is the dominant one, which is what? 0.6. Okay, so 0.6 square. If you're looking for A, then you do Q square. What is Q over here? 0.4. Now, if you're looking for heterozygotes means capital A, small a. So, for that, you use this 2 PQ. You use 2 PQ and you replace this. So, 2 into A. What is A? It is 0.6. And small a is what? 0.4. At the end, you get 0.48. This is your answer for the question. Okay, this is your answer for the question. 41 question, the answer is 0.48. Those of you who gave any other answer, it's totally fine to do it wrong. But always remember, there is a formula that you have studied in mathematics, that is a plus b whole square, which means a square plus b square plus 2ab. So 2pq is equals to 1. This is the equation for Hardy-Weinberg principle. Okay. So here you have to replace. If you want to find homozygous AA, you have to look for this. If you want to find homozygous small a, you have to look for this. And 2pq is for that. Okay. Clear? So what is your answer? Your answer is this. Now you can ask me if at all you have any doubts in your list. Whatever chapter it is, let's go. And those of you who have not clicked on the like button, on the subscribe button, it's high time to do so. Click on that, okay? Click on the subscribe button and stay tuned for all the updates. And tomorrow be there sharp at 4 o'clock because we will be having a quick revision for all the chapters entirely. So my dear children, any doubts that you are having for any single chapter, ask now. I'll be of help. Otherwise, we will wind this up and meet tomorrow directly. What say? 100%. Thank you, Anand. So Anand is definitely going to be there. Thanks, Da. And yes, as I said, no doubt, bunch of thanks, ma'am. Thank you, Monisha. Wishing you all the best. Do you have your exams today, my dear children? How many of you have your exams today? If at all you have, there is a video on in every single chapter that I have done, I have given one shot, and the questions that have come from those chapters, please make sure you're watching that in 2x speed at least, but definitely. So tomorrow's session would be about everything, like from the start of all the chapters which have been coming around. I'll try to take 10 MCQs, and I'll make sure I have some questions in that, long form questions. Okay, the most, frequent ones which are being asked. Okay? Preetam, I have my exam tomorrow. Then definitely I had my exam today. Kumada, hi, how was your paper? Oh my God. And thank you for replying for the homework question out there. And Alan, thank you so much, dear. You're a great teacher. You make me want to listen to bio lectures. Thank you so much. Tomorrow English, ma'am, and Wednesday bio. Okay, wishing you all the best. I'll be coming tomorrow, so yes, we can meet. Okay. Santosh, ma'am, human health and disease. What about it, dear? I have solved MCQ questions over here. Do you want anything else from me? Most probable questions or things like that. So tomorrow I can bring that for you guys. As I said, whatever you have in your head, please let me know now only, that I come prepared for it as well. And those of you who want to leave, please, you can leave the video as well, since, as I said, I'm listening to this to get basic understanding for the future. Awesome. So proud. Kumada, your exam, I hope it was fine only. And my dear children, all of you, 3 marks question. Monisha is saying 3 mark questions you want now or tomorrow. I can bring it to you for tomorrow, okay? 2 and 3 marker questions. We will bring it tomorrow. We will bombard on it and we will do it. Fine. Be there sharp at 4 o'clock and share it with your friends as well. Tomorrow I want all of you guys to be here itself. Okay? And my dear children, please do not be worried about anything. First chapter diagrams tomorrow. Okay, so I have the chapter diagrams video coming today itself. Probably it will be live by 8 p.m. Okay, it will be there, so please watch it. It is going to be helpful. And apart from that, I have solved a question paper of the previous year midterm. It is also going to go today. Watch that video as well. Okay, sample paper. It's a previous year question paper for your midterm examination. I have solved it fully, so please watch it. It can be of your help. Fine. Chalo, then I will leave, and you guys please take care of yourselves. Do not be worried about anything out there. You are going to do awesome, and please pray to God before you write your examination, and be fine. Yeah, that's all. So no, Anand, I won't be coming live today, as in the video would be there by 8 p.m. or something like that. Okay? Okay then, children. Wishing you best of luck, lots and lots of love from ma'am, and do not forget to hit on the subscribe button and on the like button if at all you think this video was nice. Okay? Chalo, bye-bye. Take care. See you soon. Bye-bye, guys. Bye-bye. Monisha, don't be worried. You would do good. Just keep on practicing. Tata.